Done, sir.
Yeah. Thank you. Rather putting the entire title name in the in the title, what we can do, we can give team 2 manuscript and date.
Okay, sir.
That would be great.
And like,
Okay.
yeah.
So
Done,
what
sir.
document you have shared, I am sharing it to you also. So title. Targeting Marburg virus. So, diverse plant means how many plants we are including?
Also, the like different species of citrus. And
Okay.
like,
Can we make a like instead of diverse plant? Can we make some name with citrus?
okay, of citrus plants. Like lemanoids are particularly present in just citrus.
Yeah. So something with citrus derived something. Okay. You
Okay.
think over alternative names, like what are the names possible?
Okay, sir.
Yeah, I'm highlighting here.
Yes, sir.
A structure-based drug discovery approach. Okay. Again, this part I'm thinking should be replaced with some other word.
Okay.
So whatever have to be replaced, I'm highlighting in yellow, and what are the
Yes,
possible
sir.
suggestions I'm giving in red. Okay.
Okay, sir. Yes, sir.
And I'll share the change document.
Yes,
Lemon
sir.
occurring phytochemicals. Actually, an abstract should include all the parts. Like it should have few lines for introduction, few lines for methods.
I tried to write it in that way. Like I first mentioned are like ligands. Then I mentioned the virus we are targeting,
Hmm.
and that the crux of the study is that we are studying the potential antiviral effect of these ligands on this virus, and we are
Hmm.
doing it through an in silico method. We'll be like performing the docking, and like we'll be performing the MD simulation after that.
Yes. Until here, it's fine. Like introduction, as you said, is there. In silico approach performing MD. Have we went directly? Okay. Do we went directly to molecular docking? We have not done anything before.
Like exactly like but so
See, we just told, right? That is not our only aim. You remember, we discussed in the last class.
we have to,
See, last class, what are the suggestions I gave?
last class we were like actually told to perform all the docking for all that, and like to perform a MD run, and like to make all the changes in the manuscript. And we like suggested that we'd also go through like pharmacophore modeling afterwards once we're clear with the result.
Yeah, glycons we have just concluded, and whether I have told you to compare between RNA polymerases or something, I remember. For the
Also,
your
you
team,
said that you would look for like mutants on your end too. Like that's why we shared the PDP ID. And like we mentioned that when we did from our end, like there's no mutants that we have found or like any mutants of Marburg in general.
whether this guy Ajay belongs to your group, Ajit or
No,
Ajay,
sir. No, sir.
right? Okay, I told him to look at other things. Fine, this is fine. And again, while using in silico, always remember you should utilize ice.
Italics. Yeah. Sorry.
Same way everywhere. And this you can see, right?
Yeah.
This you have to include.
Okay, sir.
Yeah. Then including Marburg virus, Ebola virus. So where are the references?
At the end, sir.
Here
We have
you
like
are.
added the numbers here,
Uh,
but we have like written them at the end.
okay. Here I hope that this references are reason one. Have you added the bibliography?
Yes, sir.
Where it is?
So let's go above. Actually, these are like the things that we forgot to remove. Here.
Ah, fine. So what I'm checking here is that all reason one, some at least about 2024, we should have some many papers.
We try to like, yeah, some of them are like very old too, like the ones that mentioned the initial things first.
Yeah.
And then we try to keep as many as we could find related to like till 2025.
Okay. Fine. Just a minute. Give me a minute.
Yes, sir.
Am I audible?
Yes, sir.
Yeah. So yeah, this one only I want to know. Like 2024, I can see some two articles only. In 25, two articles. Okay. Where you can
Okay.
add these recent versions means in the general terms.
In the introduction.
In the introduction, we have lot of scope to add
Hmm.
reason articles and that to high good impact articles.
Yes, sir. I'm like trying to look for articles that are like higher number of citation and all to
Higher number of
include.
citations and with uh, of course higher number of citations will be a good journal and reason one. Okay.
Yes, sir.
Because before going to the editor, there will be some team who are checking these basic things.
Okay, sir.
This you ensure. Then moving on. For
I like
introduction,
that.
yeah, for introduction, we don't want subheadings because the subheadings
Okay.
will be used only for subheadings will be used
Abstract.
only in no subheadings of introduction will be used only in review papers.
Okay.
So here generally we won't use it.
We just like remove them.
Yeah, we will write in the paras. But
Okay.
the structural insights and all you can okay like that you can give for your results and discussion.
Okay.
But I appreciate the way you went. Uh, research objectives and again you just thought of a thesis kind of way.
Okay. Yes, sir.
And there is another way of doing. Like if you are giving one to introduction, it should
Okay.
be two 2.1. There is no 1 1.1. Whereas here we should make two.
Hmm.
These are small things only like 2.1. Okay.
Okay, sir. Yes.
Same way you can edit this.
Yeah. We can do that. We like do the same for the results and discussions too.
Exactly. 3D. Here 3D should be there.
Okay, sir.
Mm, okay. So you have to start writing for trajectory analysis. Admin.
Yes, sir. Actually, we did admit. So the thing with the MDS run is like I was hoping if Mr. Nalapu could be here in the meeting too. Like the results that he shared with us after doing the MD run on his side are like all, very
I do not know.
not
I can hear your voice. I echo. It's my mistake or your mistake. Come again with slow, please.
Sir,
Yeah,
I'm
please
audible now?
help. I put my headphones also.
Okay.
Yes, please no.
The thing is, like, I was hoping if Mr. Analapko could also be here on this meeting. Like, he did the MD simulation for one of these complexes as like a trial. Like, he mentioned to you that he has access to a
Yeah,
high
right.
performance
I
computer.
remember. I remember
So
that
the
he
thing
said.
is, yeah, but like the results he shared on the drive, they are like, very, when I like compiled the temperature XVG or pressure XVG, it is only like showing for a very few number of picoseconds, like 5 to 7 picoseconds.
Oh my
And
God.
like the RMSG, SASTA and all, they're just not showing any graph. The values are like very low, and it is saying that when I search it up, it is saying that it is perhaps the compound is not within the
If you
5
have got the trajectory, you got the XTC file.
Yes, so I think I have. Like, I tried to, he shared the results after the HTC, all the temperature and everything. And I like tried to download them and perform them on my system. So I wanted to ask him that if he has anything else he could share, or like if he has results on his end that are maybe correct. Because like he
I do
shared
not
the
know, like
files in the drive,
whether he
but he
is
hasn't like, hmm,
aware of how to do things, or even though he attended the class, maybe when he listened to the recording, he may rethink. Like he want to share at least one data per 1 nanosecond at least to have a
yeah, so
practice.
he like, yeah, when we were originally like doing it during the MD simulation classes,
Hmm.
the run for that was done by Tanveen and me. Like we only did it for the presentation.
Okay.
And he said that he did the run for I think 100 nanoseconds, but like the result are just like for a very few short number of picoseconds.
Mm-hmm.
So yeah, that's why
See,
like if
there
he
will be two
had those,
things. Either he might have not shared the proper file to you, or he not conducted it.
okay,
See, actually, the XTC file will be in GBs. Is
yes
that
sir.
how much? 1.5 GB or 2 GB? Is that like that?
No sir. Let me just check if I can
I
give it.
think it's only a few KBs. Like I also
Then
saw.
no way, no
Yeah,
way. It should be in GB at least, or
it's
more
just a
than
few
MBs.
KBs or MBs. Yeah,
Okay,
that's what
okay,
I'm
okay.
saying. The files are not proper, so we did not include the MD simulation results.
Okay. But okay, fine. However, we will run then. If he have MD with by himself, you may save some money. That's my concern. And we also can try in different variants. But in such case, if he's not having, we have to move on with our neighborman team.
Yes, sir. That's why I was hoping like if we could talk today regarding this, but like yeah,
Definitely, definitely. See, it's a part of your thing,
yeah.
but they have not mentioned that they have not. We do not know who are going to continue, right? For that
Yes, sir.
purpose. And since you guys are sincere and you are producing results, we will proceed to the next stage. Okay, next stage in
Yes,
full
sir.
fledged, not for namesake.
Yes,
And we are planning for some good impact paper in a hi-fi journal. Hi-fi means at least Scopus Indus journals. Okay, that's our aim. So we should work towards that. So
yes, sir.
journals are expecting minimum 100 nanoseconds or 200 nanoseconds.
Yes, sir.
We can start with 100 and we can later on extend it.
Yes,
Always have a practice of giving serial numbers. Okay.
okay, sir.
Yeah, these and all you can align, I guess. What is
Yeah,
it?
I'll
You'll
add to
do.
the
Yeah, you please do that. Add
yeah, I'll
serial
add the
number.
serial number column. Yeah.
Yes. Yeah, this you should add and same way
So we'll
here.
add here too. Yeah, we'll add here too.
So have you guys prepared until the level of this one? So you just write at least the names.
Okay, of the top
Uh,
compounds.
Yeah, that
Yeah, I
is
have like
ligands bound with.
Ananya, like we can also just discuss the confusion we were having with the top binding affinity ones and the admit results.
yeah, we can mention like, can you like explain the admit results properly for those? I think we have mentioned them in writing though.
Yes, yes. So, in case of the admit results, so there are two compounds which show uh, zero Lipinski violation. One is isolimonic acid, uh,
Yeah.
which is uh, which is also blood-brain barrier permeant and is has high solubility and good bioavailability score. And the
Hmm.
second one is limonin, which also
Hmm.
has a good bioavailability score and high GI absorption. And
Okay.
just one confusion I had was
Hmm.
uh, there is isolimonexic acid, which had a good binding affinity of minus 9.5.
Where it
So,
is now like
uh,
isonymous, this one. Yes. Okay.
No, sir.
yeah,
Like
no,
the
no, sir,
top two
not this one.
compounds for binding and the top two from like admit are like very different.
Fine,
Yeah,
fine, no problem. We have to focus upon binding. This admit is a supporting data.
Okay, sir.
okay, okay.
This
Then
admit
it's
is a
fine.
supporting data. Wherever it is also orienting with this, then we can double confirm it.
Just one confusion, sir. Like
Yeah.
the second top performing ligand, which is trihydrodenine B,
Uh,
the below one, this
uh.
has two Lipinski rules violations, and has a poor bioavailability as well.
Yeah, this
But
we'll write in the discussion. Even though
Yeah,
they
okay,
are
we
having
can.
even though they are having this, the limitation of using this as it is, or so and so like that, we can write as a discussion.
Okay, we can like write
So
a separate paragraph for the limitations.
limitations, but if it is not supporting overall, we can move this to just a supplementary table.
Okay.
There's a way,
okay,
like
sir.
whichever is not directly supporting our claim, we can move them as a supplementary table and put out of the manuscript. But we'll add additionally. So this
Okay.
is not like it is not enough. Like we will keep on adding more tables.
Hmm.
See, along with RMST, RMSF, we also want to perform hydrogen mode.
Yeah.
We have to perform hydrogen bond, and we have to perform MMGBS, PBSA.
Yes, sir.
Okay, and we also can look at different torsion angles.
Hmm.
Okay, we can look for torsion angles, and even for a modeling results. So okay, that's fine. I understood regarding dynamics. What about the modeling? I taught you how to take the interaction data, interaction figures.
Yes, sir. I have like one test image that I think is going to proceed with, but I like wanted to show you first that if this is better.
Please
Uh,
show that you have now.
yes, sir. I'm just opening it.
Yeah, I just stop sharing. You please share.
Sir, I had one doubt.
Yeah.
Like one the top the ligand molecule with the top binding affinity, which I mentioned, was Kaihat B.
Hmm.
So for this one, actually, I searched through like research papers also and took the help of ChatGPT also. So it mentioned
Okay.
that since it has multiple violations and it has a very complex structure,
Hmm.
so by using QSAR,
Hmm.
we can optimize the structure further. So, like, will it be all right to perform QSAR in this
Yes,
regard, or
yes.
should we just
No, no, there is no
okay?
limitations. You can extend the study in any way. And overall, we have to bring a story. So
Okay,
what are
sir.
the tools you will be using for QSAR?
Sir, last time, like I had practiced it in my college, so right now I don't properly remember it, but
Hmm.
I need to search more about it.
Yeah, you
So,
search it, like you check for what kind of tools. If not, I will help in that tool names also.
okay, sir.
Fine.
Yeah.
Sir, is my screen visible?
Yeah, it's visible now. Yes.
This was like how I initially did it. I
Hmm.
mainly like kept all the interacting residues from the protein with one color and
Hmm.
like the ligand and opposite color, and like I made the surface to be like 80%
Yeah,
done.
this is
And
looking good. Having black background in this case is looking good. Yes.
yeah, I think this is again black. I took one in white background too.
Yeah, compared to that, it's okay, but the background is looking so good to
Yeah,
me.
that's what I was thinking. But then I was like, since maximum journals have like white background image, so I took like with white too.
Yes, good. Okay. The only thing missing here is the labeling.
Yes, sir. I have like an issue with labeling. Like the thing is, the labels sometimes hide behind the structure. So I've not
Yeah,
like
that's
found a
why.
way
That
yet
is for
to.
your understand. That is for your way. Okay.
Hmm.
Like you want to remember which ligand is lying where or which residue is lying where. For that purpose only you have to use PSC. Generally, we will edit using paint.
Okay.
Do one thing. Take a copy of it. Like close it. Copy this image somewhere. Always we should have some backup. Okay. Yeah. Copy and create a new folder like pics or something inside the same. Yeah. Anywhere. Open in Paint. Either you can write in Paint or the better way is you can open a PPT and keep on adding the pictures over there.
Okay.
That would be a better way. Even rather than Paint, PPT would be a better way.
Okay.
I think
So it was
this
like
black background also is because of transparency, I guess.
yeah.
You have not
It's
made
like
black background.
it was black. It was like I
What
forgot what the
happens
spelling
no?
is.
You copy that black background, put in your Word file, it will transform into white because it's a transparent image.
Okay.
So if you want to create a black, just paste some randomly, you can understand.
Okay.
You getting the
Like I
point?
remember for yes, I remember like I changed the background specifically to white for the third image.
Yes,
I
ma'am.
do
Like
it up.
you take some black background image and paste if you want, and this is also fine. You have to crop it. But I expected the whole image and this image together.
Which sir? Which image together?
I'll put one. Okay, if you stop sharing, I'll share you one thing.
Yeah. Yeah. I've stopped sharing, sir.
Yeah, yeah. I'll share your previous batchman's image. Hope it's visible.
Yes,
Yes,
sir.
sir.
How it is?
Yeah, it is looking good. Like it's the entire protein.
It's they shown the entire protein too, and they also clear about the interaction type of interaction. Actually, this was asked by the reviewer. Okay, so we have included that. We made specific colors for this. Okay, here that guy not changed because he took two different proteins, so he changed the color of the protein.
Okay. Okay. Okay.
But in your case, if your protein is same, only the ligands are changing. You can follow this way.
Mhm. Yes, sir.
I'll put
So
this
at
image in
least
your
like
group.
okay.
Yes. Getting the point?
Yes, sir.
Yeah. So this should be done.
Yeah, I'll try doing it like this way.
Hmm. Got it? Now you can share your
Yes,
screen.
sir.
Yes.
Yes. One second. Mhm. Yes, sir. This is the image I took now with like the black background.
Okay. You better crop it. Do one thing, copy, cut and put in. You cut this and open a PPT.
A PPT.
Yes.
But next, sir. You're not audible, sir.
I meant that copy the same in the PPT. PPT.
Yeah, I did, sir.
Now you can crop it. The advantage of having in PPT is we can add more images and we can also add we can align it to the maximum.
Okay. Is this better, sir?
Yeah.
So I
Okay.
like need to
Expand
move
it. Now what you can do, you can zoom out. Zoom out your PPT.
of the PPT.
Press control. Yeah. Little bit, little bit. Now you expand the image. Hmm. Always don't expand crosswise. Expand in the corners so that it will make it uniform. Yes. Yeah, that's the correct way of expansion. Yes. Expand again. You can go out of the box. Nothing wrong. Yes. Keep it side. Also, how many ligands you are going to arrange?
Come again, sir.
Like this is one complex, right? So far we are going
Yes, sir.
to make three complexes, correct? So
Yeah, for the MD.
yeah, three complexes you have to keep side by side.
Okay, sir.
Fine. Now what you can do, go to insert.
Go to and say.
Yeah. And type the word art. Word art, yes. Yeah, you can
Okay, I
add
think like
a normal black color one and you can convert that into white.
okay.
type the ligand name, residue name. Find the residue names. Yeah. Now you got an idea?
Yes, sir.
You can complete this now.
Yes, I
Once
can.
you complete, I'll help you.
I like do this for all the, I think the top 10 compounds that we have.
Yes, how many complexes we have?
We have a total of, I think, around 35, maybe.
Yeah, not 35 we need. If you make for 35, we can keep it in supplementary. Okay. But at least top 5 should be kept inside the manuscript.
I like add them to the manuscript,
See,
the
yes.
top 5.
So whatever we are proceeding towards MD.
Hmm.
So out of 35, we have to take at least top 10. And in the top 10, we have to proceed further to MD.
Yes, sir.
See, generally we are showing the table. These figures are an additional support, like how good they are.
Okay.
No, it's no need to mention every thing of them. Okay. And what
Yes,
about
sir.
the ligand? What about the ligands alone?
The images of the ligands.
Yeah, images.
That's it. Do we need to put the ones like we need to open the ligand in Pymol and take that, or do we like just take the 3D structure from Pumpkin?
I think like I can also draw it on ChemDraw if it's okay.
Yeah, wherever it is looking good, take it.
Okay.
Okay.
Make it something what classy, like looking the images should be very good.
Okay,
We can like
so
put the 2D and the 3D structure together.
yeah,
And,
like two separate columns, right? With the ligand
yeah.
name, PubChem ID. Mhm.
Yeah,
Yeah,
we can like make a table of just the ligands.
I'll do
Okay.
one thing.
Yes.
I'll show some basic things like others manuscript. You will have an idea. This one for your understanding, I'm putting in your WhatsApp chat.
Yes, sir.
Hope my screen is visible, ma?
Yes, sir.
Yeah. So in their manuscript itself, uh, can you see the graphical abstract? You guys also have to
Yes,
make the
sir.
graphical abstract.
Yes, so also about that, like I've been like looking at graphical abstracts of like other papers. So a
Hmm.
lot of times the use is just like straight up the images of the compound. So it's not a lot of writing done in the graphical abstract.
Okay.
Like what is like the general things that you need to like ensure that your graphical abstract has?
Fine. So what's your point here?
Also, like I want to know like what all things do we need to include in graphical abstract? Because like the ones that you showed us from the previous manuscripts, like this one, there were a lot of like the method and the structure and everything like processed
Yeah. That you
together
can
with like
you can
writing.
make it later, but you can start preparing. Like what is our focus? Okay. You
Okay.
can have some idea. But look at the 2D plots, what they have. I'll show that also. See, it's like a sculpturing. Okay. We have to edit each and every line. And
Yes,
what are
sir.
the ways we can improve? So here they made 3D and 2D like this. I won't be happy. I'm not happy with the 2D structure, but 3D is fine. Okay, like that you can have an idea.
Okay, sir. Yeah.
This one I show, and I'll show another manuscript. Here they took images from camera, I guess.
Yes, actually.
Okay, they took images as it is. At least they are looking uniform. Some uniformity should be there.
Yes, sir.
Here they have that, and few other journals. Can you guess what they compared here?
These are like the lick plot interactions and like the pi mole interactions.
Yeah. No, the right side is not pi mole. So left side is pi mole, that is Lick plus output, and the right side is Discovery Studio.
Discovery Studio. Okay, yeah.
Yeah,
So
something like
I
that.
also have like Discovery Studio. Should I also like to
Like
compare it
you try to
in
include and make many combinations, ma.
Okay.
You try to make many combinations and try to understand which combination is looking good. Look at their way of showing. You got the point?
These are, I think, yes, sir.
Yes. That's what I'm saying. Is that possible?
Yes, sir. We will try. I try to like make as many possible combinations as I can.
Ah. If you do that, then it will be so great.
Yes, sir. I also like downloaded Pymol into my Ubuntu for this so that I can like take images without the
Yes.
watermark. I try all the variations that you've shown and like look for the best.
Yes. And I will also suggest you to have Anaconda. Hope one of the class I told how to use Anaconda, correct?
Yes, sir.
Yes,
Because
sir.
after having Anaconda, we can make multiple images. Okay, we can use R programming, Python programming.
Hmm.
Okay, so that have to be done. Clear?
Yes, sir. Yes, sir.
And I want the interaction table, and I'm eagerly waiting for these images. So that will be
Okay.
happening only after doing MD. So if you submit your
Yes, sir.
MD, we can ask them to initiate the process. Before that, you please check with Mr. Onalapo whether he is able to run or not.
Yes, sir.
Okay, double confirm with him,
Yes, sir.
right? If he able to run, then no fine. If not, we have to get their help, get the help of neighborman and running. After this only, we will get these kind of images, information like energies, and we
Yes,
also
sir.
having this is the thing, free energy landscape.
Yes, sir.
And after that, hope you know about this. This is called PCA.
Yes, sir.
Principal component analysis,
Okay.
right? And these
Yes,
are the
sir.
things to be done. Majorly, what I feel is your introduction have to be improved further. Like you made that was also. I want some images, something.
Yes, sir. I was just wanted to like show you this one so that I would know like which format should I follow for all the images.
Yeah, about the virus, okay,
Hmm.
and why it is important, something related to the
Yes, sir.
work, not very generalized one related to the work. And units, try to put the units. For number, no need. But here you have to put kilocalorie per
Yeah,
hour.
for band. Yeah.
Yeah, other things fine. Like once you're done, I'll say a few more suggestions. See, we have to work hard till we publish it. Okay. Even after
Yes, sir.
submitting to the manuscript, we will have work. Hope you know that.
Yes, sir.
Okay.
Yes, sir.
Could you please fill all this with images and send to me again?
Yes, sir.
I'll rename this.
I like change the name on the drive for this.
Yeah. So in my manuscript folder, I started for team 3. So 25 team. Yeah, team 2, right?
Yes, sir.
Yeah. So I am putting manuscript today's date. So whenever you send me again, okay, I'll put like this. Team 2, manuscript. What's today's date?
Yes,
15.
sir.
We are giving on Independence
15
Day.
August.
Yeah. So I'm expecting 3 days,
Yes, sir.
right?
Hmm.
So in such case, you can not only for you, wherever you can see, I'll put name, date of manuscript. So that we'll have an idea whose manuscript it is and when they edited last. If you are resubmitting on the same day, okay, what I will do, I'll put version 1, version 2. Clear?
Yes, sir.
Okay, ma. If any doubts are there, no more doubts, we can wind up the session. If any doubts, I am ready to clarify.
Sir, can I like show you a paper that I read? Like it had a portion called the analysis of the structural changes in the complex, where throughout the run they apparently like studied the changes in the uh, residues using PDB sum. So, like I
Oh,
want to
in my
know like
paper. Yeah, PDB sum is between protein and protein. That cannot be
okay,
used for protein ligand.
okay,
It is between protein and protein
uh,
interactions.
then,
Fine. So, but
yeah,
we have lot of other tools like Discovery Studio. You can explore Lick Plus, Lick
mhm.
Plot. You try to install those.
Okay, sir.
If you start installing that, definitely will get good pictures. And look at other docking papers as you are doing right now. Look at other docking papers and try to improve the clarity.
Yes, sir.
Fine.
Hmm.
Yeah, I am sharing now. Wish you all the best.
Yes, sir. So this
Yeah.
paper has like the protein bound to capsaicin from chili, where they
Sorry, come
use
again.
PDB
Where
sum.
this?
The one that I mentioned, where they use like PDB sum for analysis of structure change, they have like the protein, TNF, bound to capsaicin.
Yeah. In such case, they might be looking for interatomic changes.
Okay.
See, the PDB ID themselves you can check. You're getting my point? Like if you go to a crystal structure,
Hmm.
I'll demonstrate.
Yes.
So it will give the interactions between any crystal structure. Can I see these structures?
Yes, sir.
Yes, sir.
See, mainly you look at here. Protein.
Mm-hmm.
Okay. Both the proteins only we used to do. But I do not know whether they have increased the way of doing. These are the characters we'll get, used to get.
Yes, sir. Yeah.
Right? Since our
Yes,
thing is an
sir.
intracellular protein,
Mm.
it will go and bind with the RNA polymerase directly. At least if it is
Mm.
outside, we will look for other parameters.
Okay.
Here, again, if you are looking for ions something, they are showing for Lick plot. They are asking you to go for Lick
Like,
plot. Okay. So they have a
yes, sir.
calcium with them. So they are asking, they are diverting themselves to Lick plot. So try to install Lick plot and check. You know how to take images in Discovery Studio?
Yeah.
Yeah, 2D plot you know to take, right?
Mm-hmm.
I'm expecting the same also. Will you do the same?
Yes, sir. Yes, sir. 2D plots from Discovery Studio and like the images of visualization and PyMol.
Very good, very good, very good. Good. Okay. Then what else?
That was all from my side, sir.
Yeah. So try to
Sir,
complete
for
and be in touch. Yes.
yes, sir. Sir, for QSAR, can I reach out to you tomorrow or maybe
Sorry
someday
for
after?
QSAR?
Can I reach out? Can we reach
Yeah,
out to
yes, yes.
you
See,
tomorrow
you
or
can text
sometime
me.
after?
You can text me in the group anytime. You can tag me and text me. Whenever I'm seeing, I will definitely come back. Okay.
Okay, sir.
I'll
Okay,
try to
sir.
sort out the issue in the mobile itself. If not, we will arrange a meeting separately.
Okay, sir. No
Yeah.
worries.
Yes. Now my concern is each time when we are meeting, we should have some data to discuss. Okay. So in such way,
Yes,
if you
sir.
are working hard and within 2 days gap, if you are sending the results also, I'm happy. I'll make a meeting immediately. My thing is we have to communicate this paper successfully. Clear?
Yes, sir.
Yeah. Okay. Thank you so much. Bye.
Thank you, sir.
Thank you. Happy Independence Day again.
Thank you, sir.
Bye bye.
Bye, sir.
Bye bye.